Hello students, welcome back to another learning lessons in our Physio-Pulse channels. In the first lecture, we have discussed how urine is concentrated through the counter-current multiplier mechanism. But today we will discuss about counter-current exchanger through the Vasa Recta and even also role of ADH and to maintain the concentrational gradient and some applied physiology. So these lectures we will discuss in a simple, conceptual and exam-oriented manner, specially designed for undergraduate students. So let's get started our today's lectures. So first of all, counter-current exchanger mechanism, as we know, the Vasa Recta functions as a counter-current exchanger, and it is responsible for the maintenance of medullary gradient, which is developed counter-current multiplier. In this figure, it is clearly shown that 300 milliosmol, then it comes to 320 milliosmol, and also 375 and 700. In this way, it can be exchanges. So role of Vasa Recta in maintenance of medullary gradient, if you see Vasa Recta acts like the counter-current exchanger because of its position, and it is also U-shaped tubules with a descending limb, hairpin vein and the ascending limb. Vasa Recta runs parallel to the loop of Henle and its descending limb runs along with the ascending limb, descending as well as the ascending limb runs parallel to the ascending limbs and its descending limb and therefore loop of Henle and its ascending limb also runs along with the descending limb. So in this way, it can exchange some ions. So therefore, the exchanges mechanism, due to this exchanging mechanism, so osmotic pressure also maintains. The sodium chloride is absorbed from the ascending loop of Henle and enters into the medullary interstitium, and from here it enters in the descending limb of Henle of the Vasa Recta. So simultaneously, water diffuses from the descending limb of the Vasa Recta into the medullary interstitium. The blood flows very slowly through the Vasa Recta, so a large quantity of sodium chloride accumulates in the descending limb of Vasa Recta and flows slowly towards the ascending limb of Henle. By the time of the blood reaches the ascending limb of Vasa Recta, the concentration of sodium chloride increases very much. So this causes the diffusion of sodium chloride into the medullary interstitium, and at the same time, water from the medullary interstitium enters the ascending limb of Vasa Recta, and this cycle is repeated. So in this way, a countercurrent Vasa Recta can maintain the medullary gradient. So students, I think you all have understand about how counter-current multiplier or Vasa Recta acts as a counter-current exchanger mechanism. So this recycling of like the ions like the urea also occurs in the Vasa Recta from medullary interstitium along with the sodium chloride. Urea also enters in the descending loop of Vasa Recta when blood passes through the ascending limb of Vasa Recta, and also urea diffuses back into the medullary interstitium. And now, role of ADH, how ADH is acts here. As you know, ADH is the antidiuretic hormone, and that is antidiuretic hormone. It is secreted from the pituitary, posterior pituitary gland. So, final concentration of the urine is achieved by the action of ADH. Normally, the distal promoter tubule and the collecting tubule are not permeable to water, but the presence of ADH makes them into the permeable, so resulting in water reabsorption. So water reabsorption induced by ADH is called the facultative reabsorption of water. A large quantity of water is removed from the fluid while passing through the distal commuter tubule and collecting duct. So the urine becomes hypertonic with the osmolarity of 1200 milliosmol per liter. So this way role of DH has maintained this. And number mainly we have discussed how exchanger mechanism Vasa Recta acts, and secondly role of ADH to maintain the osmolarity gradient. So this is the figure which you can say easily how urine is concentrated here. So this is the cortex portion, this is the outer medulla portion, and this is the inner medulla portions. So they are in this way. Urine is also this is the diagrammatic representation shown here. Role of ADH in the formation of concentrated urine. ADH can increase the permeability of water in the distal commuter tubule and collecting duct. And here the mechanism of formation of dilute urine that it can shown in these two figures. Now, very important part that we will discuss now, that specifically the summary of the urine concentrations. How urine concentration we have learned today, that if we discuss in one by one, that you will find in this slides also. So Now, see, the summary of the urine concentration is that when the glomerular filtrate passes through the renal tubules, its osmolarity is altered in different segments. What are the segments? As you know that in the structure of the nephron, there are Bowman capsules, proximal convoluted tubule, distal convoluted tubule, and collecting tubule are, and loops of Henle, they are also present. So in Bowman capsules, the glomerular filter collected, and that is isotonic to the plasma, and at the time it contains the osmotic pressure, osmotic gradient is 300 milliosmol per liter. Now, when it reaches into the proximal convoluted tubule, therefore the proximal tubule there is active reabsorption of sodium and chloride followed by the obligatory reabsorption of water. Now, how this is happened through this mechanism, this it can be shown in this figure here also. And if we have to see the osmotic pressure osmolarity remain the same as the Bowman capsule, in here also 300 milliosmol per liter, and it is also now isotonic to the plasma. Now, the next part is the thick descending portion of segment loop of Henle, and when the fluid passes from the PCT or proximal tubule and reaches into the thick descending segment, here the water is reabsorbed from the tubule into the outer medullary interstitium by means of osmosis. And it is due to the increase in osmolarity in the medullary interstitium, and therefore that is the outside of the thick descending tubule. The osmolarity of the fluid inside the segment is 450 to 600 milliosmol per liter. That means the fluid is slightly hypertonic to the plasma. Now, thin descending segment of the loop of Henle is that as the thin segment descending segment of the loop of Henle passes through the inner medullary interstitium, more water is reabsorbed. This segment is highly permeable to water, and so the osmolarity of the tubular fluid becomes equal to that of the surrounding medullary interstitium. In the short loops of the cortical nephron, the osmolarity of the fluid of the hairpin vein is 600, becomes 600 milliosmol. So 300 go increases and it comes to the 600 milliosmol. And when it reaches into the particularly the hairpin vein, this osmotic pressure is gradually increases to 1200 milliosmol. And so it means the solution of the fluid is hypertonic to the plasma. So in this way, thin segment or descending loop of Henle, that the reaches the osmotic pressure is 1200 milliosmol. Now, this fluid when reaches into the thin ascending segment of loop of Henle, there also, if you see, the gradually decreases in the osmotic pressure. Due to the concentration gradient, sodium chloride decreases out of the tubular fluid, and the osmotic pressure osmolarity decreases, which is 400 milliosmol per liter. And this fluid of the segment is slightly hypertonic to the plasma. Now, thick portion of ascending segment, here also in the same way, so reaction of sodium ion, it is decreases, therefore it can diffuse the osmotic pressure or diffuse the fluid and your sodium ion or chloride ion. As a result, the osmotic pressure osmolarity also decreases. So thick descending loop of Henle, when it reaches so 1200 milliosmol, it gradually comes back to the 1200 or 200 milliosmol when it reaches into the distal convertible of the DCT. So here an important function is ADH. ADH is present in the distal converter tubule. It becomes the permeable to water and resulting in the water reabsorption and final concentration of the urine. So here also osmotic pressure gradually increases and it reaches to the 1200 milliosmol per liter. The urine now becomes to hypertonic to the plasma. So in this way, urine is concentrated. So I think you understand that what I want to say that there's a 300 milliosmol, it goes to the 400 milliosmol, then it's 600 milliosmol, and whenever it reaches into the hairpin vein 1200 milliosmol. But here, what important thing is there, sodium and chloride decreases, then it gradually decreases osmotic pressure 1000 to 400. Then ultimately, when it enters into the distal converter tubule, it reaches to 200 milliosmoles. And once again, when it goes to the collecting tubule, its osmotic pressure also gradually increases. So in this way, it can maintain the osmotic pressures. So students, I think we all discussed about the counter current exchanger as well as the how urine is concentrated through these different types, different segments in the nephron. Now, one important thing we also want to discuss here, that is the applied physiology, osmotic diuresis. So diuresis is the excretion of large quantity of water through the urine. So osmotic diuresis is the diuresis that induce the osmotic effects like the glucose. It is common, that is called the diabetes, diabetes This the second type of disorder is called the polyuria. Polyuria means the frequent urination, and that means it is common in diabetes insipidus patients. When it occurs because of that, due to the dysfunctioning of ADH hormone, at the time in the renal tubule it fails to reabsorb the water, and therefore water is excreted. And the number three is the syndrome of inappropriate hypersecretion of ADH. It is the pituitary disorder characterized by the hypersecretion of ADH into the SID, and that means syndrome of inappropriate hypersecretion. And excess ADH causes the water retention and which decreases the osmolarity of ECF, extracellular fluid. And number four, nephrogenic diabetes insipidus. It is also sometimes common, and the condition is found when renal tubule fails to give the response of ADH, resulting in polyuria. And these are the various types of symptoms which we found in during this urinary concentration process. So I think we all covered in our lectures like the how urine is formation is happened through the counter current exchanger mechanism, and how what are the importance role of ADH, and some applied physiology like the osmotic diuresis, polyuria, etc. So thank you for watching this lecture. I hope this session has helped you understand the topic clearly. If you found this video helpful or useful, please like, share and subscribe to Physio-Pulse channels. And do not forget to press the notification bell so you never miss our upcoming lectures in physiology, biochemistry, or biology. So if you have any query, any suggestion, please give comments in the our comment box sections. And thank you. And we will see you again in our next lecture. Until then, keep learning and stay curious.